All right, let's talk about the Golgi apparatus now after the endoplasmic reticulum. We just finished with the endoplasmic reticulum. This is another membranous organelle. This one, that which the endoplasmic reticulum creates, gets sent on to the Golgi apparatus basically to package and export from the cell. And so this is the portion of the organelle. This is the organelle that can create more of the plasma membrane. So that's one of its functions, is to fix the plasma membrane. But these vesicles that the Golgi apparatus creates actually are the same material as the plasma membrane and can work their way, after they're released as a vesicle from the Golgi apparatus, they can work their way up to butting against the plasma membrane of the cell, then becoming one with the plasma membrane of the cell, and then releasing whatever's in the vesicle out to the extracellular fluid in a process known as exocytosis. So exo, out of the cell. As I said, it fixes the Golgi apparatus. One of its functions is to fix the plasma membrane, and to finish off, don't get me wrong, it doesn't just package the proteins, which hormones and enzymes are, but also finishes off the protein, that puts the final touches on it. One other function of the Golgi apparatus is to make a lysosome. And lysosome and peroxisomes, at the bottom of this screen, which I believe is going to get cut off a little bit, you have this information about peroxisomes, contain enzymes. They break down fatty acids and organic compounds. These are fatty acids and organic compounds that are not needed by the cell, or they're extra, or whatever, for whatever reason the cell doesn't want them. So the dialogue is to break them down. Now, lysosomes are a little bit more serious than peroxisomes. They are created by the Golgi apparatus, and they come out as a primary lysosome. Primary lysosome is not active. The enzymes are not active, which is a good thing because these are pretty serious enzymes. When they come in contact with an organelle that needs to be destroyed, damaged, it doesn't work, it's lived past its time, then the enzymes in the lysosome become active. And it's not known, these enzymes are so strong, it's not known why they don't actually even digest the membrane of the lysosome. So these are very strong enzymes. That organelle can be totally totally released from the cell, or if there's any portion of that organelle that can be reused, it can be reabsorbed. It can also be, it can also release its enzymes and cause the cell to die, so autolyse the cell. And then you have, on one side you have the portion of the organelle going all the way out, and then you have another one where you have the portion of the organelle being reabsorbed, and then the rest of it going out with regard to lysosome. So lysosomes are the cleanup people, break down large molecules, attack bacteria, recycle damaged organelles, and eject waste by exocytosis. Breaks down, with regard to autolysis, lysosome membranes break down enzymes. So you have these this opening up of the lysosome, and then those enzymes are released into the cell, which can only mean the cell is going to die. Another, I guess the second to the last membranous organelle is the mitochondria. And the mitochondria is a double membrane. You have an outer and inner membrane to this mitochondria. And the mitochondria we tend to associate with the powerhouse. This is where we get most of our energy currency, which is that molecule, that compound known as ATP. And without this mitochondria, we would not be able to function at all. It's kind of a symbiotic relationship between us and the mitochondria. The mitochondria gets to have a nice comfortable place to live, and we get to have all the energy we can use, which is a byproduct of what the mitochondria does. Mitochondria is a unique organelle. It has its own DNA, so its own little piece of DNA, and it has the has some of its own digestive enzymes. Produces our energy, our ATP, in two ways. One way via glycolysis, and the other way via the Krebs cycle. Oh gosh, I'm sorry, I'm going to sneeze. And the other way via the Krebs cycle, and we'll look at that in the next slide because it's a little clearer in the next slide. The outer membrane is smooth, and the inner membrane has folds, and those folds are very important because the activity that goes on in the inner membrane is highly charged. This is where the electron transport chain is active, and that is a high energy transference. If that didn't take place in this inner fold membrane, it would actually not, the whole thing would not function at all. It takes glucose and produces ATP. That's what the mitochondria does. And here's a kind of cartoony, more simplified version of the mitochondria. And what you have is outside of the outer membrane, glucose, which is a 6-carbon ring, is broken apart in this process called glycolysis into two pyruvate molecule or compounds, and what that is you take a 6-carbon ring and break it apart into two 3-carbon chains. That releases ATP, so you make some there, and that is done without oxygen. Then as the pyruvate goes into the mitochondria, it goes into the Krebs cycle, or the citric acid cycle. In the presence of oxygen, you start pulling off and breaking that down, and then it goes into the inner folds after you have pulled an electron off of one of the hydrogens, or off of as many of the hydrogens as you can. It goes into the inner fold, and this is where the electron transport chain takes place, and this is where most of the ATP is made, and it's made by phosphorylation of ADP, so the diphosphate. And what happens in those inner fold is that energy that's released from having that electron free, a free electron, manages to put an extra phosphate on ADP, turn it into ATP, release it from the mitochondria so that the body can use it. And about 95% of our ATP are created in this mitochondria. Some cells have a few. Cells like muscle cells have a lot of mitochondria. Cells like the heart is very much packed with mitochondria. So areas where you have a lot of need for energy, there's an incredible amount of mitochondria in those areas. And this is where ATP is created in the presence of oxygen, with a byproduct of carbon dioxide being created, and ultimately the currency that we use for energy, ATP, is the end product. The nucleus is the last organelle we'll talk about in this chapter. It's the largest one. It's the one that you most likely see when you look at a cell in a microscope. It's considered the control center because that's where you have DNA. It's made up of the nuclear envelope, which is a unique type of membrane that has 2 layers and a space in between. And the 2 layers and the space in between, you have little holes, which we call nuclear pores, that allow for communication, but mostly allow for that messenger RNA to get out. Also in the nucleus you have chromatin. This kind of cloudy, nebulous purple, dark purple, light purple area is chromatin. You do have a fluid environment in there, the nucleoplasm. In the center of the nucleus is the nucleolus, and the nucleolus is responsible for creating the ribosomes, the ribosomal units. With regard to the nucleus, that's where you have DNA. You have the fluid, which contains the nucleoplasm, ions, enzymes, nucleotides, which are the building blocks of more DNA or more RNA, and some RNA. But nucleotides mostly building blocks of DNA, and also storage, place you can store energy. Then some supporting filaments in there to give it structure. Again, the nucleoli, which in the other slide we call it the nucleolus, because whether you're talking about plural or singular. As I said, this is where you have the making of RNA, and so ribosomal RNA and then ribosomal subunits. So synthesis of those free ribosomes, not the fixed one, or not the fixed, well actually the fixed ones too, because the free can move on and become fixed on the endoplasmic reticulum. And also there are some enzymes and histones being produced in the nucleoli. In terms of the nucleol, the DNA coiled around histones, and you have this image here. This particular image talks about 3 or 4 different things that are terms to know at this point. So chromatin is the genetic material when the cell is just being the cell. And when you think about that, it makes sense. You have these chromosomes that we can see with an excellent microscope. When the cell is getting ready to replicate, you can see these. But when the cell is not getting ready to replicate, you want that DNA kind of unwound so that it can separate, and messenger RNA can come in, and proteins can be dictated, can be controlled. And then when that cell is getting ready to duplicate itself, the genetic data condenses into chromosomes, and so that they can be very, very organized, in an organized way, can move to the 2 new daughter cells. This idea of DNA is instructions for proteins. And I don't know how they've identified it, but I guess they counted them all. They're up to about 100,000 proteins that the body can make. Different proteins. Proteins tend to be the way the body actually controls what's happening on a chemical level. You have this idea of a gene. When we think of a gene, this can be a very scientific term, or this can be in layman's terms. But generally, we think of a gene as an area on that DNA that we know is coding for a specific trait. So whether that trait be multiple areas on a gene that codes for hair color or eye, we think of a gene as a something that's going to have an end product. Certainly in layman's terms we do. And with regard to genetic code, that's the language of DNA, and that is the sequencing of those complementary bases, genetic code. And then you have your nucleosomes and your histones here. The histones are the blue, and remember those come out of the nucleoli, and the double helix actually wraps around those to condense the genetic information into the chromosomes.